Team Francisella and Plague. Plan of the team present on this slide. And the Francisella taxonomy: domain bacteria, phylum Proteobacteria, class Gamma Proteobacteria, order and Psychotrichaceae, and family Franciscellaceae, genus and species Francisella tularensis. And Francisella tularensis is a small gram-negative coccobacillus with bipolar appearance, non-motile and capsulated. It can survive up to weeks at low temperatures in winter and soil and animal carcasses. In laboratory settings, Francisella tularensis appears as small rods, 0.2 by 0.2 micrometer, and is ground best at 35-37 degrees Celsius. And Francisella tularensis is a causative agent of tularemia, primarily a plague-like disease of rodents and other small animals. And the culture characteristics of Francisella tularensis: isolation is very difficult, as Francisella tularensis is highly fastidious. It needs special media such as blood cystine glucose agar and cystine heart agar supplemented with 9% heated sheep blood. And specimen ulcer, scrapings, lymph node biopsy, gastric washings, sputum and blood are inoculated onto the media and incubated at 37 degrees Celsius for 4 days, and aerobically, as Francisella tularensis is an obligate aerobe. And colonies are blue-gray, round, smooth, and slightly mucoid with small zone of alpha hemolysis. And safety precautions such as biosafety level 3 must be used to handle clinical specimens to avoid the risk of laboratory acquired infection. And Francisella tularensis is a small gram-negative coccobacillus with bipolar appearance, non-motile and capsulated. And by chemical properties, it is weakly catalase positive, oxidase negative. Yes, this oxidase negative and catalase positive, and maltose and it produces acid but not gas from glucose, maltose, and mannose. And direct fluorescent antibody tests can be done with commercially available antisera directly from the culture colonies for subspecies identification. And the epidemiology of Francisella tularensis. Francisella tularensis is found widely in nature and has been identified in almost 250 wildlife species, including more than 50 species of arthropods. There are several potential vectors that transmit Francisella tularensis to humans and wild rodents, and logomorph are natural reservoirs of this agent, and humans may become infected through various routes, including direct contact with an infected animal and consumption of infected food or water, inhalation of infected aerosols, or through the bite of an arthropod such as ticks and deer flies and mosquitoes, fleas, lice, midges, and or mites. Only rarely it is transmitted from person to person. Tularemia has been reported in people living in rural areas in endemic regions. If campers or hikers stay in these areas, they may get infected. And trekking, hunting, or fishing may put a person at risk for tularemia in endemic areas, and activities in contaminated water are other risk factors. Tick-borne tularemia is rarely reported, but it is probably underestimated or not identified among hikers or campers. And pathogenesis of this disease, Francisella tularensis, enters the respiratory tract and the lamina propria of the respiratory bronchioles via M cells. Digest antigen is taken up by dendritic cell. The dendritic cells travel to regional lymph nodes and present Francisella tularensis antigens to T helper 1 cells. And the T helper 1 cells proliferate. They may return to site of initial infection. Restimulation by local antigen presenting cells results in interferon gamma production and macrophage activation. And the failure to clear the Francisella tularensis results in granuloma formation. And interestingly, it has been noted that a strong innate response involving interferon gamma and the tumor necrosis factor are important for controlling bacterial replication in macrophages in the early stages of infection. And this makes sense because recall that these two cytokines are normally responsible for activating macrophages to produce reactive oxygenated species and nitric acid, which are highly toxic to bacteria. The humoral response production of antibodies is less important for elimination of this facultative intracellular pathogen. And the clinical manifestation, Francisella tularensis subspecies exhibit a broad host ranges. Disease caused by the species is subdivided into several forms based on clinical presentation, namely typhoidal, systemic signs of sepsis, and pneumonic, pulmonary symptoms, and oculoglandular air involvement, and swollen cervical lymph nodes, and pharyngeal and ulcer glandular, cutaneous ulcer, and swollen lymph nodes, and the glandular, primarily swollen lymph nodes with no other localized symptoms. And the incubation period is roughly 3 or 5 days, and the signs and symptoms people develop depends on how they are exposed to tularemia. Possible symptoms include skin ulcers, swelling and painful lymph glands, and inflamed eyes, sore throat, mouth sores, diarrhea, or pneumonia. If the bacteria are in health, symptoms can include abrupt onset of fever, 39 degrees Celsius, chills, headache, muscle aches, joint pain, dry cough, and progressive weakness. And the abdominal sonogram of infections individuals can show small pockets of ascites in the abdomen and the pelvis, containing neutrophils and lymphocytes and macrophages. And people with pneumonia can develop chest pain and difficulty breathing, blood disputum, and respiratory failure. Tularemia can be fatal if the person is not treated with appropriate antibiotics such as streptomycin or gentamicin. However, getting vaccinated is a better way to prevent an infection caused by this species. And the laboratory diagnosis of this disease, tularemia disease, is culture method. We use gram stain method and biochemical identification of bacteria, and direct fluorescent polymerase chain reaction, and the enzyme linked immunosorbent assay. And the polymerase chain reaction assay has been used to detect Francisella tularensis specific genes encoding the outer membrane proteins. It can also differentiate subspecies. And antibody detection is the mainstay of diagnosis, as isolation is difficult. Agglutination test, latex and tube agglutination, and the enzyme-linked immunosorbent assay formats are available. And next is Yersinia, taxonomic kingdom Eubacteria, phylum Proteobacteria, class Gamma Proteobacteria, order Enterobacterialis, family Enterobacteriaceae, genus Yersinia, species Pestis, Enterocalytica, and Pseudotuberculosis. And Yersinia pestis Morphology of this pathogen. This is Yersinia pestis was isolated for the first time by Alexander Yersin in 1894 in Gangong, and it is the agent of plague and fulminant systemic zoonosis and transmitted from rodents by arthropod vector. And Yersinia pestis is gram-negative oval coccobacillus, often shows bipolar staining. Ends of the bacillus stain darker than the central part, and pleomorphism, variable size and shapes in order cultures. It is non-motile and encapsulated. And the culture characteristic of Yersinia pestis on blood agar and the McConkey agar. And culture characteristics you present on this colonies of Yersinia pestis present on these petri dishes. Yersinia pestis is aerobic and facultatively anaerobic. The optimum temperature for growth, unlike most pathogens, is 27 degrees Celsius, but the capsule develops best at 37 degrees Celsius. It is not fastidious and grows on ordinary media. Various media used are as follows: blood agar colonies. Colonies are non-hemolytic and dark brown, pigmented due to the absorption of the hemin pigment. And McConkey agar, lactose non-fermenting, colorless colonies are formed. Nutrient broth, granular turbidity occurs with surface pellicles formed later. Nutrient broth with oil or with oil characteristics, stalactites-like growth occurs hanging down from the under surface of the oil. And the culture smear and the motility testing, gram staining method we used, and gram staining of culture smear reveals pleomorphism, coccoid and the coccobacillary, filamentous and the giant forms, and the involution forms are seen in older cultures. And biochemical properties, Yersinia pestis shows the following reactions: sugar fermentation, it ferments glucose, mannitol, and maltose with the production of acid but no gas, and lactose and sucrose are not fermented. And it is catalase positive. Yes, it is catalase positive and oxidase negative. And indole urease and citrate tests are negative. And Voges-Proskauer test is negative. Biotyping is done based on glycerol fermentation and nitrate reduction. And epidemiology of this disease, Yersinia pestis plague. Epidemiological factors: reservoir wild rodents such as jebel, field mice, and bandicoot found in forests as the main reservoirs of infection in India than the domestic rats. And the source of infection are infected wild rodents, rat fleas, and the cases of pneumonic plague. And vector: rat flea is the commonest vector of Yersinia pestis, which acquires infection by feeding on infected wild rodents, and several species of rat flea may act as vectors, such as Cynostyla astia and other species. And the human flea may rarely serve as vector. And the plague cycles: plague exists in two natural cycles: domestic cycle and the wild cycle. And domestic cycle, it occurs between humans, rat fleas, and rodents, and the wild cycle, it occurs in nature among wild rodents independent on human beings. And the mode of transmission: human plague is frequently contracted from bite of an infected rat flea, most common, and direct contact with tissues of infected animal rodents, and droplet inhalation man-to-man from cases of pneumonic plague, and bite of an infected human flea, and blocked flea in a blood meal. The fleas suck about 0.5 milliliter blood containing 5,000 bacilli from infected rodents. In the gut of flea, the bacilli multiply enormously and may block the proventriculus. Such blocked flea eventually dies as it cannot obtain a blood meal. And however, while making efforts to suck, it regurgitates the blood mix bacteria into the bite, thus transmitting the infection. Infection may also be transmitted by contamination of the bite wound with the feces of infected fleas. Partially blocked flea is more dangerous than a completely blocked flea, as it survives longer inside burrows, maybe up to 4 years in certain species. And the extrinsic incubation period is the interval between the flea acquiring infection through blood meal and becoming a blocked flea, which is usually about 2 weeks for xenocelia. And next is plague outbreak is likely to occur in places having Chiopis index of more than 1. Seasonality, plague is seasonal in North India, and in South India it occurs throughout the year, which may be attributed to the climatic conditions of south favoring the rodents to breed. And innate immunity subversion by Yersinia pestis. And this is neutrophils are a very early line of innate defense and inflammation at infection sites. It was commonly accepted that Yersinia pestis is eliminated effectively when phagocytosed by neutrophils, but survives and replicates when phagocytosed by macrophages. It has now been shown that Yersinia pestis grown at flea temperature, close to that of the host skin, at which the T3SS is not yet upregulated, can survive and replicate within human neutrophils. And Yersinia pestis can also transit from this first niche to a second one. Then macrophages phagocytose infect neutrophils in a cleaning process named efferocytosis. Within macrophages, Yersinia pestis can survive in autophagosomes, and it has recently been shown that Yersinia pestis is also capable of inhibiting phagosomal maturation by perturbing the host endosomal recycling pathway, allowing bacterial intercellular replication. And efferocytosis is accompanied by secretion of interleukin-1 RA, interleukin-1 receptor antagonist, a cytokine exerting anti-inflammatory effects by blocking interleukin 1 receptor 1, and signaling with strategy would allow Yersinia pestis to reduce innate immunity mechanisms and multiply, meanwhile upregulating its T3SS. T3SS is type 3 secretion system. Then its target organs, Yersinia pestis, would preferentially inject Yersinia autoproteins in neutrophils by specifically recognizing their surface complement receptor 3 and resists neutrophils toxic effectors through the activity of the lysozyme inhibitor IVY and the two-component regulatory system outer-membrane protein regulator ENVZ. And how Yersinia pestis disseminates from skin flea injection sites to lymph nodes was unknown, and it was suspected that it was suspected that phagocytes could carry the bacteria. This Yersinia pestis spread from the skin is very rapidly achieved in the absence of interaction with host cells, suggesting therefore that bacterial transport to the draining lymph nodes can be driven solely by lymph flow. And intracellular carriage by dendritic cell and monocytes motivated principally by sphingosine 1 phosphate driven hemotaxis toward lymph nodes was also reported that probably occurs more slowly. And clinical manifestation of this disease bubonic plague and pneumonic plague and the septicemic plague. Bubonic plague it is the most common type transmitted by the bite of and infected rat flea. Bacilli pass through the local lymphatics to reach the regional lymph nodes where they multiply. Incubation period is about 2 to 7 days. The onset is sudden and is characterized by fever, malaise, headache, and painful lymphadenitis. And the buboes regional lymph nodes appear as tender swellings called buboes, and the most common site being inguinal. And the butt can also be axillary, cervical, or submaxillary, depending on the site of the bite. And the children are most likely to present with cervical or axillary buboes. Bubonic plague cannot spread from person to person, as the bacilli are located up in buboes, and without treatment, dissemination occurs, leading to pneumonia, secondary pneumonia, and meningitis. And the pneumonic plague. Primary pneumonic plague results from inhalation of bacilli in droplets expelled from another person or an animal with plague pneumonia. Incubation period is short, about 1 to 3 days. The onset is sudden and is characterized by fever, headache, and respiratory symptoms, productive cough or hemoptysis, and dyspnea and chest pain. Though pneumonic plague is rare, it is highly infectious and highly fatal. Agent of bioterrorism, aerosolized Yersinia pestis is a possible source of bioterrorism attack, especially in non-endemic regions. And the septicemic plague, primary septicemic plague is rare, expected for accidental laboratory infections. Secondary septicemic plague is more common. It develops from spread of bubonic or pneumonic plague. Incubation period is about 2 to 7 days. Massive involvement of blood vessels results in hemorrhages in the skin and mucus, which may lead to gangrene of the affected site. Hence, disease was named in the past of Black Death. And the laboratory diagnosis, we use gram stain, Weissen stain, culture method, and motility testing, and biochemical reaction, F1 antigen detection, enzyme linked immunosorbent testing, polymerase chain reaction, animal inoculation we use. First, specimen collection. Depending upon the type of plague, the specimen collected are bubonic plague, pus or fluid aspirated from buboes, and the pneumonic plague, sputum and blood, and the septicemic plague, blood and the splenic aspirate post-mortem. And the transport medium, Carrier-Blair medium, can be used if delay in transportation is expected. And the direct microscopy, gram staining reveals presence of pus cells and the gram-negative oval coccobacilli with rounded ends surrounded by capsule. And Weissen stain, this is gram stain, this is Weissen stain. Or methylene blue staining. Weissen stain or methylene blue staining demonstrates the typical bipolar or safety pin appearance. Two ends are darkly stained with clear central area. And this is motility testing. Gram staining of culture smear reveals pleomorphism and pleomorphism coccoid, and Yersinia pestis is non-motile both at 25 degrees Celsius and 37 degrees Celsius, in contrast to other Yersinia species which are motile at 25 degrees Celsius and non-motile at 37 degrees Celsius. And the virulence factors of invasion, adhesion, and attachment and invasion locus, and serogrouping we use, serotyping, and etc.